Hello everyone. In my previous recorded lecture, I discussed about the principle of nucleic acid hybridization as well as southern blot, which is one of the formats of nucleic acid hybridization. This time, this recorded lecture will focus on the remaining hybridization assay formats that include northern hybridization, dot or slot blots, microarray, and in situ hybridization. So now let's start with northern hybridization. Northern hybridization, also called northern blot technique, is a modification of the southern blot technique. This was designed to investigate RNA structure and quantity. Although most northern analyses were performed to investigate levels of gene expression and stability, the analyses were also used to investigate RNA structural abnormalities resulting from aberrations in synthesis or processing, such as alternative splicing. Now, let me introduce to you the overview of northern hybridization. The first step in a northern blot is to extract or isolate RNA from the sample. The RNA molecules are then separated according to their sizes using a method called gel electrophoresis. Following separation, the RNA is transferred from the gel onto a blotting membrane. Although this step is what gives the technique the name northern blotting, the term is typically used to describe the entire procedure. Once the transfer is complete, the blotting membrane carries all of the RNA bands originally on the gel. Next is hybridization, wherein the membrane is treated with a small piece of DNA or RNA called a probe. Following hybridization, the probe permits the RNA molecule of interest to be detected from among the many different RNA molecules on the membrane. So let's now go over the major steps of northern blot technique. Let's start with RNA extraction. Obtaining high-quality intact RNA is a critical step in performing northern blot. RNA should be isolated or extracted from clinical samples in an RNase-free environment to prevent the degradation of RNA. Although there are a great number of protocols, techniques, and commercially available kits that can be used to isolate RNA, they all share these common attributes. So these attributes include cellular lysis and membrane disruption, inhibition of ribonuclease activity, deproteinization, and recovery of the intact RNA. And then after the RNA extraction process, extracted RNA should be quantified using either spectrophotometric and or fluorimetric methods. Then, the next step would be gel electrophoresis. The purpose of this step is to separate the RNA molecules by size. Agarose may be used at concentrations of 0.8 to 1.5%. For viral gene expression assays that involve smaller RNA transcripts, polyacrylamide gels may be used. Gel electrophoresis of RNA is carried out under denaturing conditions for accurate transcript size assessment. Complete denaturation is also required for efficient transfer of DNA in the southern blot. Because the denaturation is maintained during electrophoresis, a separate denaturation step is not required for northern blots. After electrophoresis, representative lanes can be cut from the gel, soaked in ammonium acetate to remove the denaturant, and stained with acridine orange or ethidium bromide to assess the quality and equivalent sample loading. Formaldehyde, which is one of the most common denaturants used, must be removed from the gel before blotting or transfer because formaldehyde inhibits binding of the RNA to the membrane. This can be done by rinsing the gel with ammonium acetate or deionized water to remove the denaturant. The next step is blotting or transfer. Once separated by denaturing agarose gel electrophoresis, the RNA is transferred to a positively charged nylon membrane and then immobilized for subsequent hybridization. The best low-tech method for agarose transfer is by a passive, slightly alkaline downward elution. This procedure, in comparison to upward transfer, is much faster and therefore results in tighter bands and more signal. After the blotting step is the pre-hybridization and hybridization step. Pre-hybridization or blocking is required prior to probe hybridization to prevent the probe from coating the membrane. Good blocking is necessary to minimize the background problems. Next, the membrane is treated with a small piece of DNA or RNA called a probe, which has been designed to have a sequence that is complementary to a particular RNA sequence in the sample. This allows the probe to hybridize or to bind to a specific RNA fragment on the membrane. And finally, we have the detection step. So if a radio-labeled probe was used, the blot can be wrapped in plastic to keep it from drying out and then immediately exposed to film for autoradiography. If a non-isotopic probe was used, the blot must be treated with non-isotopic detection reagents prior to film exposure. So although it is still used in some research applications, the northern blot has been mostly replaced by more efficient technologies with lower sample demands. Genomic technologies such as arrays, which I will tackle later, provide more comprehensive information with less demand on RNA length. So now let us move on to the next variation of nucleic acid hybridization, which is dot or slot blots. There are actually many variations on hybridization configurations. In cases where the determination of the size of the target is not required, DNA and RNA can be more quickly analyzed using dot blots or slot blots. These procedures are applied to expression, mutation, and amplification or deletion analyses. For dot blots, the target is deposited in a circle or dot. Dot blots are useful for multiple qualitative analyses where many targets are being compared, such as in mutational screening. In this technique, the target is spotted in duplicate side by side on the dot blot. The last two rows of spots contain positive sensitivity and negative control, followed by a blank with no target. For slot blots, the target is deposited in an oblong bar. Slot blots are more accurate for quantification by densitometry scanning because they eliminate the error that may arise from scanning through a circular target. The top two rows of the slot blot gel on the left represent 4 samples spotted in duplicate with positive, sensitivity, and negative control, followed by a blank with no target in the last 4 samples on the right. The bottom 2 rows represent a loading or normalization control that is often useful in expression studies to confirm that equal amounts of DNA or RNA were spotted for each test sample. So just a few reminders about this technique. Dot and slot blots are performed most efficiently on less complex samples such as cloned plasmids, PCR products, or selected mRNA preparations. Without gel resolution of the target fragments, it is important that the probe hybridization conditions be optimized because cross-hybridizations cannot be definitively distinguished from true target identification. A negative control, which is composed of DNA of equal complexity but without the target sequence, serves as the baseline for interpretation of these assays. When performing expression analysis by slot or dot blots, it is also important to include an amplification or normalization control. This allows correction for loading or sample differences. This control can also be analyzed on a separate duplicate membrane to avoid cross-reactions between the test and control probes. So now let's move on to microarray, which is also one of the few formats of nucleic acid hybridization. In 1987, treated glass replaced nitrocellulose or nylon membranes for the production of arrays, increasing the versatility of array production. A microarray or DNA chip is a glass slide carrying hundreds to thousands of probes. Arrays are sometimes supplied with fluorescent nucleotides for use in labeling the test samples and software for identification of the probes bound with sample on the array by the array reader. Array targets immobilized on the glass slide are usually DNA, so either complementary DNAs, PCR products, or oligomers. However, targets can be DNA, RNA, or protein. Targets are spotted in triplicate and spaced across the chip to avoid any geographic artifacts that may occur from uneven hybridization or other technical problems. Furthermore, microarray technology is a variation of the dot slot technique in which the dotted material is arranged in a regular grid-like pattern, with each feature reduced to a very small size so that hundreds to thousands of probes can be placed on one solid surface, such as a glass microscope slide. This technique allows for genome-wide transcription profiling or sequence determination in a single experiment. And finally, we have in situ hybridization or ISH. In situ hybridization is also a common format of nucleic acid hybridization. In situ hybridization is simply detection of specific genetic information within a morphologic context. This specialized type of solid support assay involves taking morphologically intact tissue, cells, or chromosomes affixed to a glass microscope slide through the hybridization process. Autoradiographic, colorimetric, and fluorescent methods of detection have been applied. Evaluation of the final product is very analogous to the evaluation of immunohistochemistry and requires experience in histopathology. The strength of the method lies in linking microscopic morphologic evaluation with detection of hybridization. In situ hybridization also has its own variations. When the detection is fluorescent-based, the technique is referred to as FISH or fluorescence in situ hybridization. Newer variations of FISH are exploiting the possibilities of automation, so that is your fast FISH, and expanding the information that can be obtained in a single assay. FIXION or fluorescence immunophenotyping and interface cytogenetics as a tool for investigation of neoplasms combines immunophenotyping with fluorescence in situ hybridization. FiberFISH makes it possible to simultaneously detect and map chromosomal breakpoints. This finally concludes our topic on nucleic acid hybridization. Please do not forget the different formats of nucleic acid hybridization that I discussed. These are southern blot, northern blot, dot slot blots, microarray, and in situ hybridization. Thank you for watching this video.